Welcome to the class. The name SARS CoV2 stands for Severe Acute Respiratory Syndrome Coronavirus 2. They belong to the family Coronaviridae and the genus Beta coronavirus. As you can see in the structure, it has a lipid bilayer, then a nucleocapsid protein designated as N, along with a plus strand RNA, an envelope glycoprotein or E, spike proteins embedded in the envelope, and membrane protein or glycoprotein M. If you look at the genome, you will find there are different segments such as ORF1A, ORF1B, S, M, N, E, and several accessory proteins. If you look at the description region, that is the ORF1AB, those are the open reading frame which encode the non-structural proteins or NSPs required for replication. The 1A portion transcribes and translates for the protein polyprotein 1A, whereas 1AB is the largest gene and codes for PP1AB. Together, they are replicase polyprotein. The S, E, M, and N proteins are actually structural protein. S, the spike protein. E codes for envelope glycoprotein. M codes for membrane glycoproteins, and the N codes for the capsid proteins involved in the formation of nucleocapsid. 3, 6, 7 AB, 8 and 9B are the accessory protein. If you look at the structure of beta coronavirus or SARS CoV, which happened earlier, you will find differences in the region 3 and 8. These subtle changes make the virus novel coronavirus. It is the pictorial representation of the coronavirus 2 or SARS-CoV-2 molecular biology. The spike protein of coronaviruses facilitates viral entry into target cells. Entry depends on the binding of the protein S to a cellular receptor, which facilitates viral attachment to the surface of target cells. SARS-CoV-2 engages angiotensin converting enzyme 2 or ACE2 as the entry receptor and employs the cellular serine protease TMPRSS2 for ACE protein priming. Then, via transfusion, the virus enters the cell. Next, the uncoating phenomenon takes place where the capsid gets detached from the positive strand genomic RNA. Next, the translation of ORF1A, 1B happens, which results in the production of all the non-structural proteins PP1A, PP1B, etc., which are responsible for the further replication and transcription phenomenon. Next, the two things happen. One is the formation of the genomic plus strand RNA from the transcribed minus strand mRNA and further transcription and translation of all the structural proteins necessary for the viral body formation, such as the spikes, envelope proteins, the membrane glycoproteins, and the nucleocapsid. Then the assembly takes place inside the endoplasmic reticulum. All the necessary structural proteins combined with the nucleocapsids are involved in the ER-Golgi intermediate compartmentalization, where the structural assembly happens. Then the assembly of mature variant takes place inside the vesicles, and with the process named exocytosis, the virus leaves the cell.